detected in almost every tissue that we analyzed. And you would have read, if at all you have read research papers, you come across this term called housekeeping genes, essentially saying these genes are essential for normal function of any cell. So all these genes are always expressed in all the cell types. So we found many of those, and then we found many which showed somewhat restricted expression pattern. That means they were restricted to certain tissues or tissue types in the human body. Now, if I want to put together this entire 2 to 2.5 years work in one image, and I try to tell you what information can we gather from that, this would be it. Now, this is a cluster, a simple clustering. What it shows is all the tissues that we sampled, and each column here represents a protein or a protein encoded by a gene. And here the red marks essentially say they are quite abundant in whatever tissue I sampled them, compared to all the other tissues in which we studied these proteins. So what does it tell us? All these red patches on this map show you some of the proteins that show a very restricted expression pattern. Why are they important? Now, every time we talk of biomarkers, one of the main criteria you consider is specificity. And if you could achieve that specificity in the form of its restricted expression pattern, now your future tests that you go on to develop would be much more robust, rather than taking a protein that is globally expressed, which is everywhere. Then if you fight for specificity, it is not going to work out on a long run. So that way, this is information rich but informative, saying that if one were to pursue any marker for a particular pathology that affects a particular tissue, I would put my bet, or I would start by prioritizing some of these proteins rather than a protein that is present everywhere. So that way this image is very powerful if one were to study it. And this is there in the paper that was published, so one could go and refer. And also, because we studied both fetal and adult tissues, we could also look at some of the proteins that are developmentally regulated. That means that are expressed in fetal stage but not in the adult stage, or vice versa. So I am just showing you some of those proteins. I will show you a representative example after this slide. These were proteins that were predominantly expressed in fetal tissues but not in the corresponding adult tissues. Now, yet another thing was, because we were unbiased and we didn't have a, see, there are two kinds in research. One is a hypothesis driven question, where you say, I already have this hypothesis, I want to test it. But this was the second category where we did not go with the hypothesis. We said, let the data tell us what is in there, rather than we being biased about what we want to see in the data. So that way, if I just use an unbiased look at this map, this is a constitutive proteasome, and this is the immunoproteasome that is observed in immune cells, which are on the right hand side here. You see that many of the subunits here are enriched in only immune cells but not in any of the fetal or adult tissues. Essentially saying how protein functional complexes are formed in a cell, and how this signature can give away which proteins are most important for their relevant function in a given tissue pattern. Now switching gears, this was what was done compared to what was known as a human proteome, both known and predicted. Now, I told you many of the times in science, unconventional approach becomes a mandate. And if you go historically, even in medicine, if you go to Alexander Fleming, if he was a conventional scientist looking at conventional things, so he wouldn't have found penicillin. And if you go back to the story of Nobel laureates who won their Nobel Prize because they associated Helicobacter pylori to ulcers, you will know that they were unconventional in their thinking, because the whole fraternity was thinking why ulcers were formed and you give antacids, but these guys repeatedly saw bacteria in their biopsies, and that's how they chased it, and they finally found what they found. So in this also, because we were following practitioners of this field, there is a conventional way of doing proteomics. And in this case, we also knew the caveats of conventional way of doing proteomics. So that's why we embarked on a new approach. One saying, yes, we found evidence for most of the proteins that people know, but also we embarked on a new approach where we wanted to find new proteins that were unknown to mankind. And again, I will not go into the technical details. Suffice to say, this is not the way people study proteins today. There are only a handful of groups in the world which embark on this approach, and we were one among those groups. Now, just to show you an example, don't worry about all these pictures. If you concentrate on this heat map, this is a protein that is known. That means the research community and the medical community knows this protein and where they are expressed. But this is a new protein that was unknown until we published our study. What essentially it shows is, for example, in B cells and T cells, we see production of or synthesis of this protein, but not the synthesis of the parent protein. But this is embedded within that gene. Essentially saying, by using a new approach, we identified B and T cell specific somewhat protein, which we still don't know the function. But the fact that it shows restricted expression pattern tells us that if tomorrow someone wants to pursue, this protein should have a key function in T and B cells, but not the parent gene within which this lies. So this is again a technical detail. I said we have many examples of that sort, and some of the new proteins that we identified already in the literature, we see some of them being characterized and people are associating function for them. For example, there is a new protein that we identified which was also parallelly characterized by another group from US, which said it has functions in skeletal muscle. Now, and I will switch here before and concluding it with one category of genes, which again, if at all somebody has read the literature, you will come across called pseudogenes. By the name itself, you understand that they carry pseudogenes, that means they are false copies of genes. Again, we demonstrated through this study that many of the genes that people are calling as pseudo are in fact coding for proteins. Until today, nobody was studying the function of these simply because they carried this label pseudogenes. And put together all these categories, we have identified nearly 200 proteins that were not known to mankind until this paper came out. So essentially saying there is still scope for us to understand new proteins that are produced in the body and what they do. And this is again a summary which you can find in the paper. Ultimately, the consolidated data from all this was published in Nature in May, and this was also featured on cover, which we are very proud of, because two parallel studies came out, one in collaboration with US, Johns Hopkins and India, and another one from German group, which together mapped the human proteome. That in a nutshell is the study. And now, making it relevant to the audience here, how can we leverage this comprehensive data set? I told you we unbiasedly sampled proteins across all these tissues, and we found patterns, we found proteins that are specific to certain tissue types. How can somebody go to this data and take advantage of this? Now, all of you here will be familiar. There is adult hemoglobin with these subunits and fetal hemoglobin with these subunits. Now, if you look at the hemoglobin locus, now these are the subunits that are known, and you have these pseudogenes in the middle. Now, one carries a tag called HBM, but none of the community until now could identify the protein product coming out of this particular gene. What was remarkable was we identified many peptides, short fragments of this protein, and when we looked at the expression pattern of that, that was pretty restricted to just fetal tissues. If you see here HBM, and it is expressed only in fetal tissues and placenta. Now, it was totally almost absent in all the adult tissues and also the blood cells that we profiled. Now, what does that say? This is coming from fetal tissues, and when we looked at the literature, there was one paper in 2005 that characterized that as a newly discovered human alpha globin gene, but all their efforts to identify the protein product failed in 2005 because the technology was not sufficiently evolved. So they characterized this, they said that this is a new subunit of hemoglobin, and we were thrilled to look at this because in 2014 we are talking about a new subunit of hemoglobin, which we would like to believe we have understood it well, right? So now we are doing a lot more work on this just to demonstrate that stoichiometry in what ratio it is there with corresponding to the other subunits. So this is one way you can leverage this data set and still find some of the findings that are missing in the medical literature or biology literature. And also this data set is a great resource for biomarker discovery. How? Now, if I take any disease, you need markers starting from risk assessment for non-invasive screening of early stage disease, detection and localization of the disease, even for disease stratification and prognosis, and even to monitor response to therapy and screen for recurrence. So you need molecular markers for most of these, and in some of the diseases you already have examples of these. But otherwise, many of this is done by imaging. Again, molecular guided imaging is still something which people aspire for. So that way proteins are a great choice to study to fill this niche area which requires huge amount of knowledge for all the diseases for us to stratify and diagnose. And if you just look at the FDA approved biomarkers, most of these protein markers are routinely used in the clinic for many of these clinical use. So essentially saying protein markers have been there and people are continuously using them. So understanding the human proteome is essential for us to increase this repertoire and also to give it to diagnostics laboratories so that they can add to this list and give better patient care. And even some of the markers as therapeutic targets, if you look at HER2 or B2, which is a target in breast cancer, BCR-ABL, again a fusion protein which is a target in CML, mutant EGFR, a target in lung cancer, BRAF, a target in melanoma. So there is a lot of activity and there is a lot of interest in understanding markers that show specificity as far as diseases are concerned so that you can detect them as well as use them as targets to treat them. So I will conclude, and also before I conclude I will tell you, within that large heat map that I showed you, we have observed patterns where there are some proteins that show very restricted expression pattern among these tissues, and some of these we know from literature are detectable in plasma. So that means these markers are detectable in circulation. So if someone were to now investigate some of these markers in the relevant pathologies, it's possible that we will have markers that can be assayed in plasma and be taken into clinic for routine diagnostics. Now I will acknowledge the huge international team that was part of pulling off this effort, and also the team at Institute of Bioinformatics in Bangalore. So every year 5 to 6 people from here come there, visit us, they work for about a month and they interact with students and faculties there, and I would like to see more of this in the future. And thank you, and I'll be happy to take questions.
Thank you, Doctor. Ladies and gentlemen, we understand that this is a truly remarkably a world-class research. I think it truly deserves a big applause for its wonderful work. I was also told by our colleagues that this was done even less than 1/100th of the cost of the expenditure done by the Western countries. That truly deserves a big applause. I'm sure, Doctor.
So one thing that India definitely teaches you, and all of us as researchers require, is, now people say necessity is the mother of invention. This was echoed in the previous talk also. You find a means. When you are constrained by many things, you still find a way to get it out. Yeah, of course, even now nobody is believing it. That is the part of it. International community, nobody believes that we pulled it off in that cost. Yes, it was 1/4000th of a cost of the Human Genome Project. Yeah, something
really,
like that.
really good. Now, can I request to have a question or two from the audience, sir?
Yeah.
Please, any questions? Yes, sir, please.
Achievement, I think. Oh, sorry. Hi. So I think it's a very stupendous achievement, but in terms of profiling your gene expression, one of the things that people have wondered about is how reliable or how consistent the patterns of expressions are. From a technical perspective, how confident are you guys that these actually represent the native protein expression within any particular tissue?
That's a very good question. The precise challenge in either mRNA or protein expression is it is dynamic.
Right.
See, the DNA world, when I say genome, it is static. That means I have the same DNA in every single cell and it doesn't change. The sequence does not keep on changing except for some exceptions which we will not go into.
Correct.
But this is dynamic. That means in any given time point, I take a snapshot of what is the expression pattern. Then that is not a constant in that tissue if I sample it in some other circumstance. However, having said that, now that is why you have to control in your experimental stage, when you in your study design stage, what you want to address. And your reliability on the answers come from how well you design your experiment to address the specific question because of the dynamic nature of gene expression pattern.
Correct.
So that way, this is a baseline expression pattern, reliable. However, if you tell me when a person consumes sugar, now what will happen in pancreas? Now that has to be designed in a certain way to look at the dynamic behavior of protein.
So my question was exactly addressed towards that. How did you guys control for the baseline? What were your criteria for that?
Okay, so here the theme was not necessarily to say This is exactly how proteins are expressed in all these tissues that we sampled. First thing was to see which proteins are expressed in which tissue, but under what circumstances we have not addressed in this study at all.
Okay.
So that has to be done in specific studies, which we are doing in certain circumstances, but this study did not address that at all.
Okay.
Yeah.
Was this technique able to isolate also the different isoforms of different proteins?
Yes, so we found evidence. I didn't show any of that because that was not necessarily relevant for the audience. We did identify many protein isoforms.
Okay.
So not to say we have the broadest coverage of isoforms, because it has its own set of challenges, but we did identify, but there is more work to do in that area. Yeah.
Thank you.
Yes, sir. General Nair, sir.
Yeah. Thank you.
My question is, the 30-odd tissues that you took were supposed to be normal, post-autopsy tissues. What was the audit on the normalcy of this normal tissue, number one?
Yes.
Because even if there is an occult malignancy somewhere, we know cytokines have effects on neighboring cells.
Yes.
So you can have sympathetic changes.
Yes.
And epigenetically manifested changes,
Yes.
which can be phenotypically seen at some point of time. So when I say it's normal, I have to have a robust sample to say it's normal.
Yes.
So what was your audit on that?
So, okay. So again, I would like to emphasize, we still do not want to stress upon this was the normal, the way, because it is very hard to define a normal, as you precisely pointed out. Many of these were head injury cases, and we went into we tapped into rapid autopsy programs that were running in some of the institutions for us to collect these wherever it was not a disease as a cause of death for some of these individuals. However, here the message is, if somebody goes in there saying, okay, there was no pathology associated, this was purely normal, because we haven't addressed completely a normal, that particular question, and also we haven't addressed ethnicity and population differences also, because these samples come from heterogeneous contexts. So that way it was normal histologically when a pathologist looks under a microscope, but all the other influences that would have had on the tissue, we couldn't address that. And it is also hard to address if we were to sample these many. Yeah.
Any more questions, sir? Thank you, Dr. Gowda, for the wonderful lecture.
Yeah.
We really enjoyed it. Now, on behalf of the organizers, I request to accept a small token of appreciation. A moment, please.
On behalf of the fraternity of AFMC, I request Dr. Harsha Gowda to please take a token of appreciation.
We also request the chairpersons to present a token of appreciation to Mrs. Vasumati Sriganesh, the chairman of the QMed Foundation from Mumbai, who conducted a workshop yesterday. We request her to come up on stage and receive it.
Thank you, ma'am. On behalf of the Student Science Society of Yonkers Medical College, I request Medical Cadet Archita Makaria, Student Secretary, Student Science Society, to please present a small token of appreciation to both the chairpersons of this session, Brigadier R.C. Kumar and Brigadier G.R. Joshi. Today, ladies and gentlemen, you shall bear witness to the finals of the Symposia Illuminati 2014. Here, the top 5 teams out of a total of over 30 teams will be battling it out for the government title of the Ultimate Champions. And the teams who have made it through the finals of the Symposia are, in no particular order, Armed Forces Medical College Pune, Srimati Kashibai Navli Medical College Pune, BJ Medical College Pune, Prabha Institute of Medical Sciences Pune, and VIMS Sholapur. The finals of the symposia will be starting after a hiatus of 10 minutes. We request the audience to please bear with us. Thank you.
Ameya Mazumdar from SKNMC.
Smriti Sharma from AFMC. Sugand from Gyan Sagar Medical College.
Rini Joseph from AIMS Bhopal. Dakshani from Adi Chunchun Nagiri.
Manisha Anil from MGM Navi Mumbai. Ashwita Ravi from JIPMA.
Anmol Sharma from AFMC. Chandrima Naskar from Calcutta Medical College.
Anurag Ganda from AFMC. Vedashri from Father Muller's Medical College.
Madhurima from and Sonu Kumar from
Bhamar Medical College. Karen Abraham from PIMS. Meghna Reddy from Gandhi Medical College. We also have with us the finalists of the debate. We have Prerna and Sanjana from Medicity, Hyderabad.
Abraham and Shubhra from PIMS.
Arjun and Adrita from KPC, Kolkata.
Jacob and Shikha from MGM Aurangabad.
And last but not least, Abhishek Chauhan and Akshat Kehta from AFMC. We also have with us the finalists for the quiz that's going to happen tomorrow. We have Depanjan Bhattacharya and Abhishek Seal from Calcutta Medical College.
Abhishek B. Jain and Anish R. Jain from BJMC Pune.
BB Aisha Patan, Kritika BK from Belgaum Institute of Medical Sciences.
And Anurag Sachan and Suman Chatterjee from AFMC Pune. We request the audience to remain seated for the finals of the symposium.
All of the teams in the finals of the symposium are BJ Medical College Pune, VIMC Sholapur, PIMS Pune, AFMC Pune and SKNMC Pune. We request the first team to come on stage as soon as possible. We request. Audience is please requested to be seated. Audience, please, you're requested to be seated.
We welcome on stage our chairpersons for today's symposium, Colonel Yashpal Singh, Professor and Endocrinologist, Department of Internal Medicine, and the Officer in Charge, Illuminati, as well as Medical Cadet Sohan Sen, the ex-secretary of the Student Scientific Society. We request you to take the chair and guide us through the proceedings, sir.
Thank you, Compass. Check, check. Check.
Thank you, Compass. We are going to start with the final session for Inter-College Student Symposium. Let me give a brief introduction about this, about symposium, ladies and gentlemen. Usually in medicine, we concentrate on small topics or chapters in our textbooks or subjects in our MBBS career as such. A symposium actually gives us the idea of going beyond that. Taking one single topic may or may not be in our books and then looking at it from 360 degrees. So that is what we will try to do today as we go on to the finals of this event.
Good evening, everyone. This is the first of the finals we'll be having for Illuminati 2014. There were more than 30 teams who participated in the prelims of the symposium, and we have shortlisted 4 teams. They are SKNMC, BJMC. There are 5 teams: BJMC, VMNC, Sholapur, PIMS, AFMC, and SKNMC. We have 3 judges who will be judging the symposium. I am thankful to the judges for taking their time to judge this event. We have Brigadier Das, HOD Psychiatry, Armed Forces Medical College; Brigadier Srinivas, HOD Pathology, Armed Forces Medical College; and we have Brigadier Amajit, Dean Bharatiya Vidyapeeth here to judge the symposium.
I'll speak a brief word about the rules of this competition, ladies and gentlemen. I've kept the rules simple intentionally. Each team will get 15 minutes to present their symposium, which will be followed by 2 minutes in which they will ask questions by first the judges and then the audience in 2 minutes. Each team will get 2 minutes for setting up their AV in front here. So I request the team from BJMC, slot 1, to be please present here and start setting up the AV as soon as I finish this slide. So each team will get 2 minutes to set up the AV, but teams are requested to stick to the time limit, please. Otherwise, marks will be deducted from your total. So I request the team from BJMC to please start setting up, and your 2 minutes time for setting up starts now.
50,000 for heart, out of which only 0.06% get one. 25% for cornea, 1.4% unlucky enough to get a liver, 2.85% get a kidney. Merging the statistics, we realize that only 10% of these actually get the transplant. Out of the rest, 10 patients die every day. Even though India has the potential of cadaveric donations numbering to 80,000, our donation rate is just 0.8 per million per year. We, the students of BJ Government Medical College, Pune, are here today to speak about organ donation awareness.
Organ donation is the act of voluntarily authorizing the removal of one's own human organs or parts thereof for therapeutic purposes. We conducted an extensive, pre-validated and pre-structured survey approved by college authorities. It was aimed at studying the level of awareness. We got 690 responses online from 18 countries and 270 offline from medical and non-medical colleges, schools, societies and local trains in Pune and Mumbai. Adding the two, we had a total of 960 with 15% medicos in offline and 40% medicos in online.
We realized that 50% of the people did not know that heart, liver and bone marrow can be donated, while more than half the people did not know that skin and pancreas can be donated. Let's take a look at the organs which can be donated. Lungs, heart, liver, kidneys, pancreas and small intestine. The tissues which can be donated include cornea of the eye, skin, bone, heart valves, cartilage and ligaments. All these can be donated in case of brainstem death. In cardiac death, most tissues can be donated, while in live donation, liver and kidneys are most commonly donated. To facilitate organ donation, our country has laid down the Transplantation of Human Organs Act 1994. The Act discusses brainstem death and its declaration, living donation, and regulation of transplant activities.
In our survey, we found that 54% of the people did not know about brainstem death. out of which an astonishing 14% were medicos. What is brainstem death? Brainstem death means the stage at which all the functions of the brainstem have permanently and irreversibly ceased. But a brainstem dead person has a beating heart. The skin is still warm, hair and nails still grow. This is possibly why, in our survey, 40% of the people believe brainstem death and coma are the same, out of which a shocking 20% were medicos. How is brainstem death declared? There are two sets of independent evaluations done 6 hours apart by a team of doctors, including a neurologist. Now, let us look at living donation. Who is allowed to donate? The father, mother, brother, sister, children, spouse, and grandparents can donate. If none of these are a match, a friend or distant relative too can donate after approval from the authorization committee.
Thus, with the government enacting these laws, more transplants are taking place. However, the high demand and low supply of organs has led to the emergence of organ trafficking and trade. We found in our survey that even today, 35% of the people do not know that organ trade is illegal. And although the government bans commercial transactions in organs, there is proof that it still takes place.